Hello, students, and welcome to another week of problem solving session for basics in biology. This is corresponding to lecture week 2. It's already 3:05, and I see people are still joining. So, since not many students had joined at 3 o'clock, we'll wait for some more time and allow them to join. Meanwhile, I'll present my screen, share my screen with you guys. And as you all know, all the existing students who had attended the first class, my name is Ashutosh Behera, and I am the teaching assistant along with two other PMRF scholars for this course. So let me just quickly share my slides. Yeah, great. So is my slide and screen visible to you all, and am I clear and audible to you all? Just let me know through the chat box. A thumbs up or a yes or a no. Great. So to all those students who have joined recently, I would like to introduce myself again. My name is Ashutosh Behra, and I am a PMRF scholar at IIT Kanpur, and I'll be helping you guys in this course, Basics of Biology, which is taught by Professor Vishal Trivedi at IIT Guwahati. Right? So this is the format of this live session would be that there will be a set of 10 to 15 questions from the corresponding week lecture that we will be doing every week. So today we are going to solve questions related to all the chapters that was taught in week 2 week 2 of this course, if you have seen the recorded lectures. So these are exemplary question or old assignment questions which will help you brush up your knowledge and clarify your doubts and refresh your memory about the subject. These will be multiple choice question, one or many options may be correct, and I have also attached explanation slides to these multiple choice questions so that you can refer to these slides anytime. So this is the format of the live session that we'll be following, and I would request all of the students who have joined new and also to the existing students to please turn off your video and mic during this session for the smooth conduction of this session. But this session will still be an interactive live session. You can interact with me through the chat box. I'll be posting these questions and we will try to solve them together. We'll wait for a minute to collect answers from all the participants or most of the participants, and then move forward with the correct answer and the explanation. So I'll post these questions in the presentation, and then we'll try to solve these answers. And these questions will be from lecture week 2. Okay, with that kind of a brief introduction, I hope I'm clear, and I see many people are trying to join this call. So no bar in joining this call. I am adding everyone to this call. So I hope the instructions are clear, and let us begin with the first question, right? The first question is, the central core of the earth is known as dashed. What is the central core of the earth? Option A, pyrosphere. Option B, lithosphere. Option C, barysphere. Option D, atmosphere. So I would request all the students to participate and take the maximum benefit out of this live session. Please post your answers via option or the word itself in the chat box, and I'll be looking at it. Let's wait for a minute to collect some answers. These questions will kind of help you in solving your original assignments for those who have registered for the examination of this course. So the assignments do have weightage, but these are not the exact questions you'll be facing in the assignment. These are some exemplar questions. And the first question I would like to repeat: the central core of the earth is known as dashed. Option A, pyrosphere. Option B, lithosphere. Option C, barysphere. Option D, atmosphere. So no need to shy out, guys, because you do not have to turn on your video or talk to me via audio. You can just silently post your chat in the chat box, your answers in the chat box. Right? I see there are some answers. The population is really thin today. I only see 10 students might be. Ramya says lithosphere. Chanchal is saying B. Aparna says option C. Okay, so since there is a thin population today, Nandini says C. Okay, cool. Since there is a thin population today, we'll wait for shorter times and spend more times on the explanations. Right? So let me reveal the first correct answer is option C, barysphere. The central core of the earth is known as barysphere. It is not lithosphere, it's not atmosphere or pyrosphere. Okay, let us understand from inside to outside what are the different layers our earth is made up of, or any planet is made up of. So, starting from the outermost surface where we have the mountains and the oceans, the thinner outermost surface is called the crust, continental crust. And these crust, along with the outer crust, the and the inner crust, forms the lithosphere. The topmost layer is comprising of lithosphere. Then we have the next layer, which is called the mesosphere or the mantle, pyrosphere or mesosphere, which is the mantle. And then when we move deeper inside, we have the outer core and the inner core. So the inner core is solid, and that is known as the barysphere. So the lithosphere has crust, upper solid mantle. So Earth's most internal layer beneath the lithosphere. So the core lies beneath the lithosphere, right? So we have lithosphere, then we have pyrosphere, which is the mantle, and then we have the barysphere, which is the core. Although barysphere is a outdated term, which is generally referred to as the Earth core, right? It is the innermost layer of the Earth, which includes both the inner core and the outer core. So the whole core part is the barysphere, and is composed of nickel and iron. So the solid inner core is composed of nickel and iron, and it is also the source of molten liquefied metals, heavy metals, and magma. And what is the temperature of barysphere? It is very, very high. The temperature of barysphere is almost 5500 degrees Celsius. 5500 degrees. It's very, very hot. And this core is also responsible for the planet's magnetism. How strong the Earth or a planet's magnetic field is dependent on the core of the planet. Is it clear to everyone? The core, which is known as the barysphere or the centralmost part of any planet and our Earth as well. Is it clear to everyone? Then just let me know in the chat and we can move forward. Good. Thank you. Okay, so here I have another explanation of all the layers of the earth: the inner core, the outer core, the mantle, and the crust. So, in thickness, the crust is the thinnest part of the planet, and that is the area which is inhabited by all the living organisms. There are no living organisms inside the mantle or core because the temperature is really high. So the other options, let us explain the other options. The pyrosphere of the mantle describes a zone where magma forms. I was saying just above the inner core, sorry, the outer core is a mantle where there is volcanic activity. It refers to the part of Earth's mantle where rocks exist in partially molten state due to high temperatures. So the mantle is also harboring molten rocks. The lithosphere is the rigid outermost layer of the Earth. So the crust is a part of the lithosphere and the uppermost part of the mantle. It is the solid rocky layer that forms the Earth's surface and includes both the continental and oceanic plates. So it constitutes of the bottom floor of the ocean and the continental plates, which forms the mountains. And the atmosphere is the, as we all know, is the layer of gases which is above the lithosphere or the crust. It is divided into several layers based on temperature changes and composition. Okay, with that we end with the first question. Here is the second question. Which of the following is not an experiment to disprove the theory of spontaneous generation? I'll read you the question again. Listen carefully. Which of the following is not an experiment to disprove the theory of spontaneous generation? Option A, Reddy's experiment. Option B, Spallanzani's experiment. Option C, Louis Pasteur's experiment. And option D, Robert Hooke's experiment. So there are four famous scientists listed over here, and which of these scientists performed an experiment which does not disprove the theory of spontaneous generation? These questions are a bit leveled up from the first assignment that we did, which will enable you guys to think more deeply. If you remember all those experiments that the options have listed here, only then can you answer this question. Which of the following is not an experiment to disprove the theory of spontaneous generation? I would encourage all the participants, please post your answer in the chat box. I'll be looking at it. Let's take a minute to gather some answers. I see some new faces have joined. Don't be shy, take your time, think about the questions. Keep this session interactive in a two-way conversation, and keep posting your answers or your queries in the chat box. Which of the following is not an experiment to disprove the theory of spontaneous generation? It's okay if your answer does not hit the right answer. We will have a long explanation of why the correct answer is the correct answer, right? And you will finally get cleared of your doubts or your understandings. So Ramya is asking, so this question was one of the assignment question, but not sure. Yeah, so these questions are not exactly what you will face in the assignments, or you must have seen them. But if you just think about it, these are exemplar questions from the same set of lectures that you will be facing in the assignments, right? So they kind of uh, revolve around the same concepts. So question might not be directly the same. There might be a chance, but still, these are different questions. So Ramya says A and Aparna is saying option D. Are others comfortable to answer? I'll wait for a minute. Think about all the experiments, all these experiments from these scientists. Which of these does not disprove the theory of spontaneous generation? For that, you first have to know what is the theory of spontaneous generation, right? I would want you guys to be really honest about and recall it from your memory, nor and don't Google it, please. This is a live session for your revision as well. How better you have remembered or understood the lectures. Okay, there are two, three more answers. Aparna is changing her mind. C maybe. And Nandini says D. Mrs. Babu is also saying D. So meanwhile, there are people requesting to join this call. I am letting all of them in so that they can join us in this discussion. For all the new people who have joined recently, so this is the quick format, quick brief of format that we are following in this live session. Please keep your videos and audios turned off. You can interact with me through the chat box. Here is a question before you, and what do you think is the correct answer? You can post it in the chat box. Okay, let's not delay it any further. I have got 4 or 5 answers. So, which of the following is not an experiment to disprove theory of spontaneous generation? The correct answer is option D, Robert Hooke's experiment. Very good. You guys have answered very correct. It's Robert Hooke's experiment, which is not one of the experiment which disproves theory of spontaneous generation. Right? So let us look at all these experiments in detail. The first up is Francesco Redi's experiment. So what did Francesco Redi do? He took a piece of a meat, put it in the jar. Now when the jar was uncovered, then there were flies and you could see maggots growing on the meat. Maggots are the larvae of fly. So when there is an open container, there is formation of maggots in the meat. Now what he did was, he sealed the container with a cork, tight sealing. There is no passage of air, so no flies. Now when the meat was kept in a cork sealed container, there is no formation of maggots because there is no exchange of any air or gases, and the meat piece is sealed. Now then the third experimental setup was with a gauze. Now gauze is a thin cloth which allows passage of air but not passage of organisms, right? Not passage of fly or maggots. Now then he saw that the maggots then started growing on the gauze, but still the meat did not form any maggots. So this concluded that for for generation of new life, you need a pre-existing life. So Reddy's experiment disproved spontaneous generation for larger organisms by proving that maggots came from flies, not from meat itself. So the meat, which is a non-living thing by now or an object, cannot give rise to maggots on its own. The maggots come from a pre-existing living organism, which is the fly. So that is how Francesco Redi's meat and maggot experiment disproves spontaneous generation, that life cannot develop out of a non-living object. Right? So this was the first option, Redi's experiment. This disproves spontaneous generation. Now the second experiment is Spallanzani's experiment. Now what did Spallanzani do? Spallanzani is an Italian scientist, Lazzaro Spallanzani. So he did a modification of Francesco Redi's experiment. He did a gravy broth. Gravy broth contains meat and all the non-living mixture, just like a curry gravy, right? So he boiled the gravy. Now the gravy, whatever organisms were there in the gravy, they are dead because of high heat. Now he kept one of those flasks in the open, like this, and the other flask was sealed with a cork, right? And then he could see that the gravy is teeming with microorganisms, which was kept in an open flask. Where the flask was exposed, that gravy has microbial growth. But the gravy where the flask is sealed is free of microorganisms. Now, how did it happen? Because the gravy did not had any microbes to begin with, since they were all heat killed. But the air or the surroundings had tiny microbes which entered the flask and then populated this gravy. That is why the microbial growth is seen in a unsealed gravy. unsealed flask, sorry. So Spallanzani's experiment proved that microorganisms did not arise spontaneously, they came from the air. So this is the conclusion of Spallanzani's experiment. Because in the sealed flask, when there is no air contact with the broth, there is no microorganism culture. This experiment contradicted spontaneous generation and supported biogenesis, that life comes from pre-existing life, right? This is also a contradiction to spontaneous generation, the Spallanzani's experiment. Now let us look at the third experiment, Louis Pasteur's famous swan neck experiment. So Pasteur did a swan neck experiment. So there were critics which were trying to criticize Spallanzani's and Francisco Redi's work, saying that you did not allow air to pass through the container. That is why you do not see maggots. But then Pasteur's swan neck experiment did it for once and for all. So what he did was he designed a beautiful swan neck bottle, with a bottle like this, where there can still be the broth can be still be in contact with air, but the microbes cannot directly come in contact with the broth. The microbes will be trapped here in the swan neck. So first he did the same with the Spallanzani's idea. He applied heat. He heated the broth, then let the flask sit, but still there is no bacteria, because now this disproves the theory that air contains microorganisms, but microorganisms cannot reach the broth due to this design of this swan neck. That is why there is no bacteria present. Now when he removes the neck, or he breaks the swan neck, there is microbial growth, and otherwise if he tilts the flask sideways and let it sit, there is still bacteria growth occurring. So this shows that microorganisms comes from pre-existing life, not from spontaneous generation. If that was the case, then this case would also have microbial growth, right? And air alone does not create life, it must carry microbes to contaminate the broth. So air alone, which is this condition, air alone could not grow any microbe. It must carry microorganisms which need to reach the broth for its cultivation. This experiment definitely disproved the spontaneous generation and laid the foundation for germ theory, that diseases are caused by microbes. So Louis Pasteur was one of the great scientists who developed sterilization methods to free surfaces from microorganisms, as well as liquids like milk and water, right? So all these three experiments that were listed in the options, which was Francesco Redi's meat experiment, Spallanzani's gravy broth experiment, and Louis Pasteur's swan neck experiment, all three of them disproved theory of spontaneous generation. Now the fourth option was the correct answer, which did not disprove the theory of spontaneous generation, which was Robert Hooke. Now who can tell me what is Robert Hooke famously known for? I haven't included this in my slides. Can you tell me what is Robert Hooke's contribution to biology? Just let me know in the chat box. Quickly. What do you think Robert Hooke is known for? Very famous name in field of biology. Yes, you are absolutely correct. So Robert Hooke discovered he saw a single cell for the first time. He took cork cells and saw it in a microscope. So he was a guy who invented the microscope. First primitive microscope was discovered by Robert Hooke, and he saw the cells. Yes, you are correct. All the chat box comments are really correct. So, but Robert Hooke's experiment does not disprove spontaneous generation, right? Was it easy to understand? I had listed all these experiments for you guys to revise it. If it's okay, then we'll move on to the next question. Great. Cool. Okay. The third question is, Dash is an example of a connecting link between Arthropoda and Annelida. Now this is a memory based question, if you remember. Dash is an example of a connecting link between Arthropoda and Annelida. Option A, Peripatus. Option B, euglena. Option C, lizards. And option D, seals. Now, which of these do you think is an example of a connecting link between Arthropoda and Annelida, the insects and the worms? I hope you guys know what is a connecting link. Only then you can answer this question correctly. Okay, I see a flurry of answers. Farsana says A, Nandini A, Chanchal, Shivkumar, Aishwarya, Mrs. Babu, Aparna, everyone is unanimously saying A. Now, is this the total strength that we are having today? I think so. Okay. Okay, let's check what is the correct answer. You guys are all correct. The correct answer to this question is peripatus. Option A, peripatus is the correct answer. Peripatus is an example of a connecting link between the insects or the arthropods and the annelids. I would like to take back that insects are just a class within the kingdom arthropods. There are many other arthropods as well, which are not insects. But the peripatus is a connecting link between annelids and the arthropods, which are two different classes. Now, this is how a peripatus looks like. It's a worm. Peripatus is also known as a velvet worm, is a transitional organism bridging the evolutionary gap between soft-bodied annelids and hard-bodied arthropods. So it has jointed appendages, but it has still a soft body. So that is why it is a connecting link. The transition between annelids to arthropods happened through this animal, right? And it provides evidence for evolution of jointed appendages. As I was saying, it had jointed appendages, which is a characteristic of arthropoda, and a shift from hydrostatic skeleton to an exoskeleton, right? The leeches or the annelids, with the most common example is a leech, has a hydrostatic skeleton, which is a slimy or a smooth skin. That has transitioned into an exoskeleton, hard exoskeleton, like you see in crustaceans and insects or in the arthropods. And its characteristics suggest that arthropods evolved from annelid-like ancestor through gradual adaptation to terrestrial life. So now that the annelids were living an aquatic or a semi-aquatic life, the arthropods evolved from the annelids with a gradual adaptation to terrestrial life. So living on land needed hardened exoskeleton and jointed appendages to climb the terrestrial surfaces, right? So that is why the velvet worm or the peripatus is known as a connecting link between arthropods and the annelids. Okay, is it clear to everyone? Now, since we are a small group this time, I'll keep asking after every question. If it is clear, only then I'll move forward. Good. Great. Moving on to the fourth question. What is the half-life of U-238 approximately? The half-life of U-238 is approximately dashed. Option A, 5.5 thousand years. Option B, 4.5 million years. Option C, 5.5 million years. Option D, 4.5 billion years. Now, what is the half life of uranium 238? U 238 stands for uranium 238. Let us wait for a minute to collect some answers. Wow, it's good to see this response. Ramya, Chanchal, Kavi, Farsana are saying D. Aishwarya, Aparna, Nandini, Shiv Kumar, Priya, Mrs. Babu are all saying D. If you guys are saying D, then it has to be D. You guys are correct. The correct answer is 4.5 billion years. 4.5 billion years is the half-life of uranium 238 isotope, right? If you remember the lecture series, that the lectures that have been recorded by Professor Vishal, he has used this similar kind of a diagram to show the decay profile of uranium 238. Now, uranium 238 is a radioactive element. So here we see 4.5 into 10 to the power 9 years, which is 4.5 billion years. And uranium 238 decays via alpha, beta, and all different kinds of radiation decay, to give rise to different elements. It loses its mass and change in atomic number. So uranium 235 first converts to thorium 234, to protactinium 234, and the corresponding half-life of these elements have been shown on the left. 24.5 days for thorium 234, 1.14 minutes for protactinium, while again it comes to a stable molecule, uranium 234. And similarly, the uranium decays all the way to the last stable molecule, which is lead 206, with a series of alpha beta radiations. The gamma radiations have not been shown here, because alpha and beta radiation leads to change in atomic number and mass number, right? Beta is 0, 1 and alpha is 2, 2, 4. So decrease in atomic number and the mass number from alpha and beta radiation, while gamma radiation does not change any atomic and mass number, right? So uranium 238 finally decays to lead 206, and the half-life of this molecule is 4, and it takes almost 4.5 billion years for a uranium 238 to decay to lead 206 for 50% of the quantity, right? And this property of uranium 238 is used for dating fossils and rocks. The proportion of uranium 238 to lead present in any sample or in a rock sample is used for its dating, birth dating, like how old is this piece of rock or a fossil. Good. Correct answer for everyone. All good. A similar question, if you have answered this correctly, then I would accept that you have really thoroughly have a good memory and thoroughly went through these lectures. Question number 5. Potassium disintegrate to form dashed. Potassium, radioactive potassium, K40, mass number 40. It disintegrates to form dash. Option A, radium. Option B, neon. Option C, argon. Option D, xenon. This can be a little tricky. If you really have been through the lectures, you can answer this correctly. Potassium K40 disintegrates to form dashed. Is it radium, neon, argon, or xenon? Let's wait for some time to you guys, so you guys can refresh your memory and let me know what is the correct answer. I see some answers. I just hope you haven't Googled it. Meanwhile, I was waiting for all of you to answer. Aparna says argon. Ramya and Mrs. Babu are also saying option C. Okay. What about others? Can we wait for some more time? Please come up and answer. It is just for your benefit as well, right? It might happen to you that you get a similar kind of a question in your assignment, or it might help you understand the concept better or refresh your memory better, right? Potassium 40, K40. K stands for potassium. Okay, Nandini is also saying C. Okay, let's not wait any further, and let me reveal what is the correct answer to this question. Potassium disintegrates to form argon. Option C is the correct answer. Well done. So how does potassium, what does potassium decay into? Just like we saw uranium 238 decaying into lead, the final product, and in between forming many other elements, potassium can decay into two elements. One is a calcium 40 isotope, and then which can further decay to argon 40, or potassium can directly decay into argon 40. Now, these principles are used in a type of birth rating, again known as potassium argon birth rating. Now, what is the principle? Potassium is a radioactive isotope that decays into two products, as we just saw. One is calcium 40, 90% or 89% of the times it makes calcium 40, which is not useful for dating. And the other product is argon 40, which is trapped in a rock and used for dating. So the the potassium, the rock deposits deep down in the earth, when there are fossils. How to date fossils? You use these methods. The argon, the gas that forms after radioactive decay of the potassium, is trapped in those rocks, which can now be detected. The percentage of conversion of potassium to argon can now be detected as a principle for potassium argon dating. Now, when volcanic rocks crystallize, they trap potassium but not argon, which is a gas, and escapes. Cool. Then what happens? How do we date it? But over time, potassium decays into argon, which gets trapped in the mineral structure. So the volcanic eruptions release a lot of argon gases. Those are not of our importance because they escape. But the potassium within the magma deposits gets trapped and decays into argon, which is now trapped in the mineral structure. Now, by measuring the ratio of argon 40 to potassium 40, scientists can calculate rocks aging using the half-life of potassium, which is again a very high time. Half-life of potassium 40 is 1.25 billion years, but it is still lesser than uranium 238, which is 4.5 billion years. Is it clear to everyone why we call it potassium argon dating and why the answer was argon? K40 decays into calcium 40 and argon 40. Now the deposits of argon 40, ratio of concentration of argon 40 and potassium 40 is used to determining the age of the rocks, age of rocks and fossils. Is it clear to everyone? Just let me know in the chat and we will move forward. Good, great. Okay, we are moving fast with a fast pace. Now on to the sixth question. This is also a memory based question. The Cenozoic era is dashed. What is the Cenozoic era? Is it option A, 4,000 million years ago? Option B, 1,000 million years ago? Option C, 3,000 million years ago? Or option D is the present era? What is the Cenozoic era? I'll wait for a minute or so and then I'll reveal the correct answers. Meanwhile, you guys keep posting in the chat box. Okay, I see all of us have answered. That is a very good sign. And all of us have agreed upon option D, which is the present era. Option D, which is the present era, right? And I'm happy to say that you guys are really correct. The Cenozoic era is the present era, the era of humans. That is the Cenozoic period. So option D is the correct answer. Let us have a bird's eye view of all the different periods or eras that we talked about. So this is the latest era that is going on, is the Cenozoic period. So we have humans here and we have all these organisms. So before Cenozoic era, there was this Mesozoic era where we had Jurassic, Triassic, the age of dinosaurs, and they faced a mass extinction. And after that, the Cenozoic era started. And before that, we had Paleozoic era, where the fossils were found. The ammonite fossils, the amber fossils. We had mostly water-based organisms, and Proterozoic Archean era. Archean era is the start of evolution of formation of life on Earth, which is characterized by bacteria and archaea, right? So each era is depicting many periods: Holocene, Pleistocene, Pliocene, Miocene, Oligocene, Eocene, Paleocene, just separated by millions years ago, or MYA. That is often denoted for these eras. But the Cenozoic era is the human era, and as per as per the activities of humans as us ourselves, we are destroying our Earth with a higher rate, and we might face our mass extinction really soon, maybe in a million years. And a million years in the geological time scale is very short. For us, 100 years is a long time, but in a geological time scale, from the birth of all life forms on the Earth, a million years is a short time. Okay, moving on to the seventh question. Colloidal aggregates of large organic molecules are known as dash. Colloidal aggregates of large organic molecules are known as dash. A. Coercevates. B. Aerosol. C. Protocell. Or D. Emulsion. Little bit of chemistry. I'm kind of hinting to the lecture in which this was covered. Colloidal aggregates of large organic molecules are known as coercevates or aerosol or protocell or an emulsion. Let me wait for a minute and look for some answers. Colloidal aggregates, right? Okay, I see that people have answered. Farsana, Ramya, Mrs. Babu, Aparna are all saying option A. Do we have more people? I don't think we have more people. We are just 6 to 7 people today. I think 100% of the class is answering. Oh, Kavi is also here. The option A. Good to see that all the participants are answering. So the correct answer to option 7, question 7, is option A, coercervates. Well done. That's the correct answer. You guys have very good memory. So coercervates are the colloidal aggregates of organic molecules. Now this is a concept of how life originated in Earth. Life originated in Earth due to these coercevates. Now what are coercevates? What happened in a prebiotic Earth? Prebiotic Earth, when there was no life on Earth. So there was the formation of a primordial soup, as scientists call it. By soup, they mean the mixture of different components inside water. So there was first, water was a first place where life originated. So in the water, there were these molecules like proteins, there were nucleic acids, there were fatty acids or lipids, inorganic salts, and organic chemicals. These were the components, and they formed these coercivate droplets. And there were proteins, nucleic acids, lipids, and sugars and salts, all interacting with each other as coercivates. So, coercivate of a lipid interacting with a coercivate of a nucleic acid. And these interacting systems now got together and ahead of evolution to form a closed compartment known as a cell. That is how unicellular organisms came onto life in prebiotic Earth. So there is molecular exchange within the environment, interaction between different coercivates, leading which led to the theory of biogenesis, or the theory of life, the evolution of life on Earth. So coercivates are very important. Now, what are the other three options that we looked at? First is an aerosol. Now, what is an aerosol? Aerosol is a small particle or a droplet that is suspended in the air. So, in air as the medium, something that is suspended in the air is known as aerosol. Aerosols can be solid, liquid, or a mixture of both. They can be natural or human made. And the best example of an aerosol is an aerosol spray that I have shown here on the left. So this is an aerosol, any particle which is suspended in the air. Now, what is a protocell? The third option. A protocell is a simple model of cell that is used to study how life may have originated. Protocells are thought to have been the precursors to cells on early Earth. So protocell is the next stage of interaction between coercivates. Now that the coercivates have interacted, now they form what is known as a protocell, which is a primitive cell, where this interaction can be closed in and can be concentrated, can be focused. So here is an example, protocell showing a protein interacting with an RNA packaged inside a fatty acid membrane, which is the basic organization of a cellular membrane. And what is an emulsion? An emulsion is a mixture of two or more liquids that are usually immiscible, meaning they don't normally mix. In an emulsion, one liquid is dispersed in the other as small droplets. Now, the best example is mayonnaise that we eat. Mayonnaise is just an emulsion made using emulsifiers from egg fat, right? So, on the example on the left, we have oil and water, which are immiscible liquids. Oil will never dissolve inside water. Now, if you mix oil and water, what they form, there will be tiny oil droplets floating inside the water, and they will form a solution which is now known as emulsion, oil with water. So we looked at what is an aerosol, what is a protocell, and what is an emulsion, and what is a coercivate. Coercivate is an interaction between all living organic molecules. Is it clear to everyone? Just let me know in the chat and we can move forward. Great. Cool. Moving on to the next question, question number 8. The highest level of organization in cylinderates is dash. What is the highest level of organization in a cylinderate? Unicellular, B, cellular level, option C, tissue level, option D, organ system level. Coming back to the animal kingdom. Now, what are cylinderates? You need to remember what are cylinderates. Now, what is the tissue? What is the level of organization? Highest level of organization. Is it option A? Are they unicellular in organization, cellular level, or the tissue level, or at organ system level? Cylinderates. I see a lot of quick answers. I think people are familiar with the animal kingdom then, which is a good thing to know. Cylinderates are what organisms and what level of organization do they possess? People have answered option C. Farsana, Mrs. Babu, Ramya, Kavi, Aparna, Aishwarya, Nandini have all answered option C. And option C is the correct answer to this question. Option C, tissue level organization, is the highest level of organization in cylinderates. Now, cylinderates are nothing but jellyfishes. Jellyfishes uh, belong to the phylum Cnidarias, and they all belong to the class cylinderates. They show tissue level of body organization. The middle layer is also called mesoglia, acellular matrix, which is just filled with jelly. And jellyfishes do have a nervous system, a simplified nervous system made by tissues, but they do not have specified spinal cord or a neuronic ganglions, just not a brain as per such. And they have these poisonous tentacles, which they have 10 poison cells known as nidoblast, hook-like cells, which give them the name Nidaria, right? You guys are great at remembering the animal kingdom. So this was obviously a correct answer. Okay, nothing more time to dilly-dally here. Moving on to the next question. The four-limb invertebrates is an example of dashed. Now comes the good part. The four-limb invertebrates is an example of dashed. Option A, analogous organs. Option B, homologous organs. Option C, convergent organs. Option D, coexisting organs. What is the four-limb invertebrates an example, a good example of? Is it analogous? Is it homologous? Is it convergent? Or is it coexisting? Four-limb in the vertebrates. Oh, there are a lot of answers. Farsana, Kavi, Aparna, Nandini, Ramya, Aishwarya, Priya, Shiv Kumar, Mrs. Babu. Okay, all have answered. All have answered. Very good. Option B. Option B. Four limb invertebrates is an example of homologous organs. This is the correct answer. Very good. Now let us look at what are homologous organs and how does the four limb invertebrates fall into this category. Now homology refers to similar structures in different organisms due to common ancestry. So let me break this statement down for you. What is homology? Similar structures but different organisms. And why does this happen? Due to common ancestry. So there was a common ancestor in evolutionary period which now bifurcated into two different animals. Now both of these animals have similar structures, but they are now two different organisms. That is known as homology. So between these two organisms, there is homology. Now vertebrate limb is a classic example of homologous structures where the basic limb plan is conserved across different species but modified for different functions. The best example here is the human hand, which is made up of these basic organization. We have a single long bone from the shoulder joint or from the body, which is the humerus. Then we have two different bones, the radius and the ulna. Then we have a pentameric five bone structure, which is the carpals, metacarpals, and phalanges. The same goes for our toes and our feet, toes, feet, and the legs. We have a single thigh bone, two bones in the shin or in the knee joint, and then five bones in the foot or toe. Now, a similar organization is also seen in all mammals or all vertebrates. The cat, the bat, frogs, whales, lizard, and birds all have similar organization, but the function they perform is different. A human hand is optimized for grasping or holding things, while a bat hand, which is made up of the similar bone structure, is suitable for flying, while a whale flipper is functioning differently for swimming of the animal. So homology refers to similar basic body organization in two different similar structures in two different organisms performing different functions, indicating that they have evolved from a common ancestor or from a vertebrate common ancestor. Is it clear to everyone? Although I guess it's clear because you guys have all answered correctly for this question. If it's clear, then we'll move on to the next question, which is very similar to this. Now, looking at question number 10, which of the following is an example of analogous organs? Which of the following is an example of analogous organs? Option A, four limbs in vertebrates. Option B, thorns and tendrils in plants. Option C, insects and bird wings. And option D, hind limbs in vertebrates. Which of these following is an example of analogous organs? Let's wait for a minute and collect some answers. I'll just grab a bottle of water. Just bear with me. Okay. Which of the following is an example of analogous organs? Right? Technically, it should not be option A since we just saw the previous question in which it was a homologous organ, four limbs in vertebrates. So it might be option B, C, D. And let me see what you have answered. Oh, it's many people have answered C, but someone has answered option A. I think he was not there in the previous question. Okay, just let me reveal what is the correct answer. The correct answer is option C, insects and bird wings. Insects and bird wings are an example of analogous organs. Most of us have answered correctly. Congratulations. Now let us understand what do we mean when we say analogous organs and what is their analogy in between an insect wing and a bird wing. So analogous structures are body parts in different organisms that have similar functions but different evolutionary origins. Right? Let me simplify, let me break down this statement for you. Analogous structure, organisms of different origin. The ancestry is different, but the function has brought them together. They serve a same function, but their structure and ancestry is different. While in homologous organs, the ancestry is the same, structure is the same, but the function is different. Right? So, wings are a classic example of analogous structures because they have evolved independently in different species for flight, but they do not share a common ancestral origin. For example, the bat wing, which is a mammal, the bat wing has a limb, vertebrate limb kind of structure. It has a radius, the humerus, the radius ulna, and the five tarsals and metatarsals. Right? But the bird wing has a different structure. It has feathers, bones are shown in the same color as homologous. And then there are insect wings, which are totally not made up of any bone or feather, rather than, but they serve all the same purpose. The insect wing is responsible for flight, the bird wing is responsible for flight as well as the bat wing. Despite being from different ancestors, the bat is a mammal while the bird is a bird, is an avian, and insect is an arthropod, right? So these structures are called analogous structures. I hope it's clear to everyone, those who had confusion between homologous and analogous structures. Very good, very good. Okay, we have just a few more questions left. I would urge participants to answer these questions. Four or five more questions to left to go, and then we will end this session, right? So those who haven't yet answered or haven't gotten a chance to answer, please follow up with the next question. Question number 11. Which of the following organ is an extension of the integument? Which of the following organ is an extension of the integument? Option A, feathers. Option B, bird wings. Option C, insect wings. Option D, the skin. All of these are different organs, but only one of these is an extension of the integument. Now you have to know what is an integument first to answer this question, right? What do you think is the correct answer? extension of an integument. Hmm. Let me see what are people answering. This is a difficult question, might put people into confusion. I think Aparna is saying C, Farsana is saying C, um, somebody named U, I think the account, someone says A, Ramya is also saying C, Nandini is saying D maybe. Okay. What about others? Do we have more people? I don't think we have many more people with us. Which of the following organ is an extension of the integument? Okay, Aishwarya and Mr. U is saying A and Kavi is saying C. Which of the following organ is an extension of the integument? Okay, let me reveal what is the correct answer. The correct answer is option C, insect wings. So insect wings are the organ which is an extension of the integument in the insect. Now integument, what is an integument? Insect wings are extensions of the integument, which is the skin and its accessory structures in arthropods. The wings are flattened regions of the integument that form from two layers of the integument that are closely opposed. So this is the structure of the integument. Integument is nothing but the covering of the arthropod or the insect. And these coverings extend to form the insect wings. The hind wing and the forewing is also called from the thorax, right? In the insect, we have the head, the thorax, and the abdomen. Now, in the thorax part, the exoskeleton or the integument, exoskeleton has many layers. One of the layers is integument, which forms the wing. So, the correct answer is insect wings, which is the extension of integument. Okay, moving on to the next question. Question number 12. The amphibian heart has dash chambers. Amphibian heart. Option A 4, option B 3, option C 2, option D 1. How many chambers does an amphibian heart has? Is it 4, 3, 2, 1? Okay, people are saying option B. Is it option B? Amphibian. You have to. Okay, just a minute. Sorry. Okay, the amphibian heart. B, option B, three heart chambers. And you guys are correct. The amphibian heart has three chambers. So here is a whole picture that I'm showing here on the slide of all the different organisms depending on the evolutionary time and evolutionary origin and the chambers, the heart, how the heart has evolved, right? First, we have smallest organisms, which are so not the smallest, but the lowest organism in the evolutionary time scale. We have the fishes. Fishes have one circuit, two chambered heart. It has a ventricle and an atrium. It is just a one circuit from gills to the body. Then comes the frogs or the amphibians, two circuits and three chambered heart. They have one ventricle but two atriums, and there are two circuits. One atrium goes to the lung and the ventricle goes to the body. Then comes a little higher evolved turtles and lizards. Two circuits, five chambered heart. They have number of chambers increased. Then comes crocodiles, two circuit, four chambered heart. Birds, two circuit, four chambered. And mammals like us, humans, two circuit and four chambered. So we have two atriums, two ventricles. Then one of our circuits flows oxygenated blood to the lungs. There is gaseous exchange, so deoxygenated blood to the lungs. There is gaseous exchange, oxygen dissolves in, and then the oxygenated blood, red colored, is transferred to the atrium, which is now went inside the ventricle. Now ventricle pumps it to the whole body. Now the cells utilize the oxygenated blood, they release their waste as CO2, which is now carried as deoxygenated blood to the atrium, which enters the ventricle, the right ventricle, and this right ventricle pumps it again to the lungs for this cycle to repeat. Now the lungs can do the gaseous exchange again to send in a fresh array of oxygenated blood inside the body through the heart. So our answer lies here, which is frogs, two circuits, three chambered heart, which is the amphibian heart. Is it clear to everyone? I hope it is, since you guys have already answered really correctly. Okay. Moving on to the next question. Coelacanth is connecting link between dash. Option A, non-chordates and chordates. Option B, bony fish and amphibians. Option C, amphibians and reptiles. Option D, cartilaginous and bony fish. This is an organism called coelacanth. Coelacanth is the connecting link between which two phylum of organisms? Not phylum, I would say. Order of organisms. Are there non-chordates or chordates, bony fish or amphibians, amphibians and reptiles, cartilaginous and bony fish? I'm waiting for a minute to collect all the possible answers. I see there are people who keep joining and leaving this call. Coelacanth. Okay, let's have a look at all the answers. Nandini is saying option B. Mrs. Babu, option B. Ramya, Aparna, Nandini again. Okay, option B. Priya and you, Farsana, Aishwarya, all are saying bonefish and amphibians. Kavi has also answered option B. You guys are absolutely correct. Option B is the correct answer. Coelacanth is the connecting link between the bony fishes and the amphibians. So this is a phylogenetic tree showing all the related animals to coelacanth. So we can follow. This is from the evolutionary scale, from lowest to the top latest, earliest to the latest. On the top we have cartilaginous fish like sharks, right? Then we have ray finned fish like these, the stingrays, and ray finned. Then we have the coelacanth, which is this living fossil. This is also known as a living fossil. And then we have lungfish. Now all these are completely aquatic, marine. Cartilaginous fish cannot live outside water. It dies on land. Ray finned fish as such is also the same. Coelacanth is now the connecting link between aquatic to a terrestrial transition. Now, the lungfish is a fish found in muddy waters, right? Partly on the land and partly inside a moist atmosphere of a mud. It can breathe through its skin. The skin breathing is a characteristic which now gets prominent in the class of amphibians. Then we have totally terrestrial animals like the birds and the mammals. So, mammals which are at this end cannot survive inside water except for few mammals like blue whales. So, here an elephant is completely terrestrial while the cartilaginous fish is completely aquatic. While the coelacanth acts as a connecting link between transition from aquatic to a more terrestrial life. So the coelacanth is considered as a connecting link between the fishes and the amphibians. It is also known as a living fossil since there are still coelacanth found in waters and oceans. Coelacanths are lobe-finned fish that are more closely related to lungfish and tetrapods than ray-finned fish. So if you see the distribution here, coelacanths are more closely related to lungfishes. And tetrapods, the animals which move on four legs, like we have four limbs, than the ray-finned fishes. Tetrapods include amphibians, reptiles, birds, and mammals, of course. Coelacanths are considered living fossils because they are the only remaining member of a taxonomic group that is only known from fossil records. So the fossil records show the presence of coelacanth. That means they have been present from millions and millions years ago, and still there are species which there are animals of coelacanth species which are still found in water. So they are considered living fossils. They have been passed down from generations and millions of years. That is why they have been called the living fossils, and they are a connecting link between the bony fishes and amphibians. Is it clear to everyone? Just let me know with a thumbs up or a yes or no. Great. Thank you. And early paleontologists believed that the coelacanths may have been a missing link between marine and terrestrial animals. So this is what we were discussing just before. It might be a missing link between the marine life and the terrestrial life. Okay, moving on to the 14th question. Two more questions, the 14th question and 15th question, right? So try to let us try to have maximum participation. The term ontology recapitulates phylogeny was coined by Dashed. This is a famous phrase from a scientist. The term ontology recapitulates phylogeny was coined by Dashed. Option A, Alexander Fleming. Option B, Charles Darwin. Option C, Ernst Haeckel. Option D, Carl Linnaeus. Which of these scientists have coined this term? Ontology recapitulates phylogeny. Is it A, Alexander Fleming, or Charles Darwin, Ernst Haeckel, or Carl Linnaeus? Let us wait for a minute to gather some answers. There is a comment in the chat box. Sir, everybody reading for this PPT, you request this question, please. Okay, I couldn't understand what you were trying to say. Everybody reading for this PPT, you request this question. Yeah, I've requested this question, right? So you guys can answer. Maybe, okay, so whatever queries you have regarding the session and the conduction of this session and any related queries, we will take it after the question solving session, right? I'll have a 10-minute chat with all of you guys, uh, where I'm planning to solve all your issues. I think the comment meant he wants access to the presentations. I had received some requests for accessing the presentation and I have granted all of those. I firmly believe. Okay, let's focus on this question. The term ontology recapitulates phylogeny was coined by dashed. I see some have answered, some people. Mrs. Babu, Nandini, Ashwarya, Fasana are all saying option C. Any more answers? People who want to answer? There are some amount of people. People who are coming and joining by, even if you have left some, you have missed some of these questions, you can still look up the questions that I'm displaying on the presentation for you guys, and then write your answer in the comment box. Who coined the term ontology recapitulates phylogeny? Okay, let me not wait for any longer. Correct answer is Ernst Haeckel. Option C, Ernst Haeckel. Very good for all of those who have answered correctly. Congratulations. So this term was coined by Ernst Haeckel. What did he mean by that? This is a phrase that describes the idea that an organism's development mirrors the evolutionary history of its species. It is the foundation of recapitulation theory, which was proposed by Ernst Haeckel in the late 1800s. So let us understand what he meant by this. The organism's development, like this is the excerpt from his own paper and research paper and books that he published from Ernst Haeckel. Any organism's development mirrors the evolutionary history of its species. Like in vertebrates, we have the presence of these gill slits, pharyngeal gill slits, which are a characteristic of chordates. So in the embryonic stage, we can see some features, whatever feature or the ancestry of the species can be determined from the embryonic stage itself in ontology when the embryo is still developing. And at very smaller stages or very early stages, most of the embryos look very similar. You see, this is a salamander, but this is a chicken. So this is a chicken, this is a turtle, but all of their embryos do look very similar. Only at a stage they attain characteristics. They attain characteristics which make them look different or which tell us about their ancestry. Right? On the right, we have a pig and a rat and a human looking almost very similar. So, the earlier you move in development, the earlier you observe a zygote or an embryo in development, they look almost similar, but and they harbor characteristics and features which will then distinguish them according to their species and ancestry. So this is meant what is meant by ontogeny recapitulates phylogeny, and this was proposed by Ernst Haeckel. You guys did answer very correctly. Okay, moving on to the last question of this session. Question number 15. Archaeopteryx feathers are example of which kind of fossilized remains? Lots of paleontology and fossil biology questions here. Archaeopteryx feathers are an example of which kind of fossilized remains? Option A, imprints. Option B, petrified remains. Option C, impressions. And option D, coprolites. These are different kinds of fossils. Now, Archaeopteryx feathers are a example of which kind of fossilized remains? Let me know what is your choice of answer in the chat box. Someone is asking, sir, do you know Tamil? I'm sorry, I do not speak Tamil. I can chat with you guys in English, which is the formal language here, but my mother tongue is different, but I cannot speak Tamil. I'm sorry. I don't know how to speak in Tamil. Apart from that, let us answer this question. Archaeopteryx feathers are an example of which kind of fossilized remains? This is the last question, guys, so I'll urge everyone to answer. It's okay if your answer is not the correct answer. We will eventually know what is the correct answer, right? Along with the explanation. I'm sorry for the noise or inconvenience, but we are just reaching to the end. I'm just recording this from my room. Okay, since people have no idea about this answer, everyone is just trying to guess. It's fine, it's fine. You can just come up, be confident. Some are answering option B. Nandini says option C. Ramya is asking is it D. Kavi and Farsan are sure that it's B. Petrified remains or imprints or impressions or coprolites. Archaeopteryx feathers. So the correct answer is option C, impressions. This is the correct answer, guys. Archaeopteryx feathers are an example of impressions, which is a kind of fossilized remains. And what do I mean by that? So this is a fossil of an Archaeopteryx that we can see here. I've just picked it up from Google. So you can see beautiful feather impressions here. Those are not actual feathers, the feathers have degraded, but the feathers have pressed onto the soft rock while this animal was fossilized. And you can also see its bone structure, the beak structure, the eye have all really well preserved in the fossil. Now, fossils of Archaeopteryx, a small bird-like dinosaur, shows impressions of feathers and other characteristics that support the theory of evolution. Now, what these impressions, how do these impressions play important role in theory of evolution? What do these impressions show? Feathers. The fossils show impressions of advanced flight feathers, which suggests that feathers evolved before the late Jurassic period. So, even before the Jurassic period, the feathers must have evolved for the Archaeopteryx to have such feathers. So now, if an Archaeopteryx, which is such an old animal, still has feathers, then the feathers must have evolved before that. And advanced flight feathers, not just primitive feathers, Archaeopteryx was capable of flight. Then the Archaeopteryx has also reptilian features. How? The fossils show that Archaeopteryx had a long bony tail. It had teeth, claws, and belly ribs, which are also found in the reptiles. So these are some characteristics of reptiles. A jawed, uh, sharp teeth, a jawed mouth, a long bony tail, and claws and teeth for hunting, right? In reptiles. So these are all reptilian features that were discovered in Archaeopteryx fossils. And it also has bird-like features. The fossils shows that Archaeopteryx had broad wings and feathers, which are also found in birds. So this Archaeopteryx fossil gives us so much information that this animal had a reptilian feature, had bird-like features. Sorry. So this is also known as the missing link between birds and reptiles. Just like coelacanth is the connecting link between ponyfishes and the amphibians, Archaeopteryx is the missing link between birds and reptiles because it shares common features of the birds and reptiles. And it also shows advanced flight like feathers, which suggests that feathers evolved even before the late Jurassic period or the dinosaur period. Right? Is it clear to everyone? Great. So, with this, we have come to the end of our live session. We have finished 15 questions. I'll just stop the slide sharing. Yeah, so I was just yeah, I was just saying that we finished some questions related to lecture 2, lecture week 2, where you have 4 to 5 lectures upon origin of life and evolution theory. So, according to last class, I had. So, this is the time where you can ask me any questions or any doubts or any suggestions you have for these live sessions. So, in this manner, we will solve all the week, all the weeks together, following 10 to 15 questions, which can be solved in a span of 1 to 2 hours, roughly. So, as promised in the last class, I will share the YouTube link for all these videos. So these classes are recorded classes. So the YouTube link is shared with the NPTEL coordinator, and the slides that I have been presenting will also be available to you guys. You have to request the NPTEL coordinator, or you might receive it as well. I will submit these slides as well as the recording of this whole class, and this will be uploaded on the YouTube, while this presentation will be uploaded in the Drive folder for all those students who are attending these live classes, so that you can follow up with these questions and solve your assignments. Yeah, so people were asking, sorry, U23Z, I don't know, I could not figure out. For your clarification, I do not know Tamil, I do not speak Tamil. My mother tongue is different. My mother tongue is Odia, I'm from Odisha. But this is a formal platform, so we all have to communicate in English. And these recordings will be present in YouTube as well for all the students to access. Sir, assignment. So the assignment is what you have to figure out yourself. I am here to help you with the assignment or to brush up your concepts, but I will not be solving your direct assignments, right? That would not be fair for all those who have registered for the examination. You find out the answers from the lectures, you follow your concepts, then you will be able to solve your assignments very nicely. So just wait for a minute. I'll quickly paste the YouTube link in which I'll be sharing my channel link. You can follow for all the videos, upcoming videos, and the previous videos that I have recorded for this course. This is basics for biology. I'll just paste it here. That is why I urge all of the students. There were more students in the first class who dropped out, I guess. They didn't like the class, or they found other interesting things to do. But those students who are staying back in the class would get the profit of uh, these links that I will just share right now. Just wait a minute. So I have created a playlist on YouTube, which I'll be uploading all the videos related to that in a single playlist. So I'll just share the playlist link with you guys. Hmm. Yes, this is the playlist. I just copied the link address and I'm pasting it on the chat box. I'm sorry. Can you just confirm whether you can click this link and go to the channel, YouTube channel, where you can find the recorded classes, which is a screen recording for all this. Get a shareable link. I have copied this, or you just try out with this as well. So there are two links that I've shared, which contains the recorded, which will contain all the recorded lectures, starting from week 1 to week 12, as we move forward with the classes for basics in biology. And there are two other classes or live sessions happening with two other uh, teaching assistants for the same course. Yeah. So that's it for this class as well. So see you soon in the next week with lecture week 3. I hope you guys all participate and read your lectures thoroughly. If you have any doubts, you can still ask me and post me in the chat box after we solve these questions. But I'm sure that the number of questions that we try to the number of questions that we try to solve here will help us and cover us the whole assignment. And I hope you like the flow of the classes. If you have any suggestions of how I can improve or how the interaction can be improved or the quality of slides, you can feel free to text me during the class in the chat box. I'll surely read it after we have finished the session. Okay, with that, I'll end this call. Thank you. Thank you for joining. Bye bye.
Thank you, sir.